So of course, sorry, I'm extremely sorry. So Sakshi, are you there? I think you were supposed to talk before Anshu.
Hello, am I
Yes.
audible?
Yes,
Yeah.
Sakshi. Yeah, go on.
So good evening, everyone. And thank you, Aparna, for your wonderful talk. Now I cordially welcome our next esteemed speaker for today, Dr. Anshu Bharadwaj. She is one of the best computational biologists in India right now. I can say that. She is a senior scientist in a CSIR MTech Chandigarh, and she has been the lead PI in the OSDD project, that is Open Source Drug Discovery Project. And Dr. Bharadwaj did PhD in life science in CCMB Hyderabad in 2008, I think. And then she did postdoc in CRI Institute Paris as a group leader long-term fellow. So she's going to deliver her talk on a bacterial bioinformatics, and I would request to Dr. Bharadwaj ma'am to please join and enlighten us with her words. Thank you.
Thank you, Sakshi, for that introduction. Just one correction. After my PhD, I never actually went for a postdoc. The CRI one is a group leader fellowship. So I was running my own lab there in Paris.
Oh, wow. That's great, ma'am. That's great.
Yeah. And I was fortunate enough that when I finished my PhD, I was offered a job at IGIB. So that is how I started my career. One day I was a PhD student. The other day I was practically independently running. I was supposed to be running my lab, which was pretty scary. But yeah, it had its learning. So yeah, it has been
a wonderful journey, right?
Yes, I learned a lot during that.
Yeah.
Okay, now can you confirm if you can see my slides?
Yeah, ma'am, sure. Please go ahead.
So what I'm going to do today is we will learn a few tools through demonstration. And I think if you rerun the YouTube video where the slides will be available, you'll be able to do the stepwise process of how do we understand antimicrobial resistance, okay? But before I start that, I just need to know, just use this icon at the bottom of your Zoom link to raise hands and tell me how many of you had antibiotics at least once in your life.
Yes, I had Jayashree here.
Right. So exactly, just raise your hand or thumbs or something and say, okay, yeah, me. So if you can see from all the participants, within a fraction of a few seconds, everybody is raising a hand, right? Because that is something that all of us are basically surviving on. Whenever there is an infection, whenever there is a diabetic patient, chemotherapy going on, there is transplantation, surgeries. It's not just infections, but throughout the entire spectrum of healthcare, antibiotics are prescribed, right? Now, the reason I wanted to focus on antimicrobial resistance is not just to give you a training in bioinformatics or tell you what tools are to be used. I think this is a problem where all of us need to understand how each one of us can contribute in many different ways, not just as researchers, okay? But before I go into further details, for some of you who are new to the concept of antibiotic resistance, let me tell you what it is so that, you know, we have some common understanding of the terms that we are going to use downstream. So it is very simple. There's a population of bacteria, as you can see here on the slides. This is all gray, and there is one specific one which is marked in a golden color, right? Now the point is, when we give antibiotics, the ones which are gray in color here are susceptible. That means the antibiotic is able to destroy them, kill them. The one which is in golden color is something which is showing resistance to this antibiotic, right? Over time, the susceptible ones will die and only the resistant strain proliferates. So by giving antibiotics, in some sense, we are driving evolution in the direction where we are selecting for resistant bacteria. Now that is scary, but we cannot stop giving antibiotics, right? We just need to be more cautious in either prescribing antibiotics if we are clinicians or taking antibiotics as patients, right? So this is the problem. So we know, and antibiotic resistance is not there because of antibiotics. It has always been there. because bacteria need to survive. One bacteria produces some toxin which kills other bacteria. So these organisms learn to survive in this environmental niche by basically acquiring armors in which they can actually have a resistance to these environmental stress conditions. And one of them happens to be your antibiotics. Now, what I have shown you in the previous slide was like a screenshot of this, what happens. A couple of years ago, a group at Harvard did a very interesting experiment, and I would request all of you to basically go to their YouTube video and take a look at it. What they did was very innovative. So imagine, I'm pretty sure you must have heard about a Petri dish where you actually pour media and the bacteria grow on it. Imagine a Petri dish which is of the size of 4 feet by 2 feet, like a typical dining table in our homes, right? It's a pretty big Petri dish. Now in that big Petri dish, they actually poured media, and then they added color to the media, which makes it dark blue or black. Okay, and that is what you see here on the slide. It's a 4 feet by 2 feet Petri dish. The media is filled in. And what they then did, they actually demarcated the Petri dish into different sections, as you can see. And what do these sections actually say? The zeros on the edge means that there is no antibiotic. The one right in the center means just enough antibiotic to kill the organism. And as you go forward, so no antibiotics, MIC, then 10 times the antibiotics, 100 times the antibiotics, 1,000 times the antibiotics. So you would imagine the expectation was that the bacteria should not be able to go beyond 1 because this concentration was enough to kill them. What happened actually was, within a matter of less than 2 weeks, what they could see, the white thing that you see on the right hand side of the panel, the E. coli starts to appear on the panel, which is no antibiotic. And you can see that it actually starts to grow in MIC, like 1 times antibiotics, 10 times, 100 times, and also 1,000 times antibiotics in a matter of 2 weeks. Now, this is called the Microbial Evolution and Growth Arena Plate Experiment, which for the first time allowed us to visually see in a matter of days how antibiotic resistance appears and how quickly the organism can adapt. Now, how was this happening? Because bacteria were spreading through evolution. They were trying to devise mechanisms to counter the increasing concentrations of antibiotics, right? Now, we saw that it happens in lab. What happens in actual clinical scenario? What happens when you give antibiotics to people, like when we take antibiotics? So this is a slide. I just thought it will be easier to show it this way. Look at penicillin. Now, 1943, the penicillin, the antibiotic, is introduced for public consumption. But even before that, in 1940, it was already showing resistance in some of the human population. So the point was that at some point when these antibiotics were discovered, they actually started to show resistance practically at the same time or a year later or some year earlier than that, which tells you that this evolutionary mechanism happens in lab, it happens within the human host also. And because this became such a big issue, like any antibiotic you discover will at some point start showing resistance, which means that they will stop working. And that is where WHO actually came up with a definition of antimicrobial resistance, saying that if today, say for example, I can kill X bacteria with A antibiotic and tomorrow this antibiotic is not killing this bacteria, it means it is the phenomenon of antimicrobial resistance. And if there are many such antibiotics available and they are not able to kill the bacteria, then these bacteria are supposed to be called as superbugs. And this is what you see in media, print media, newspaper articles. Now, superbugs is like a day-to-day, it has become a normal vocabulary, English vocabulary, rather than something which is more scientific in nature. And that is when in 2017, WHO came up with a list of priority pathogens, saying that till the time new vaccines, new models of drug discovery or new diagnostic methods are developed for these priority pathogens, there is no other public health intervention that can save us, right? Which means that if tomorrow we get a paper cut, like sometimes your hand gets cut by a piece of paper, right? And if that gets infected and none of the antibiotics work, that cut can kill an individual. It can be that dangerous, okay? And that is why, and given that, I thought it will be interesting to show what kind of mechanisms happen. And today's talk is not to really describe mechanism, because it can be a huge workshop in itself. The whole point is that bacteria is evolving in the sense that it takes the antibiotics and throws it back, efflux. So the bacteria is not even staying in the system to act. It will change the protein structure to which the drug was binding, and then the drug is not binding to the mutated version of the bacteria. So, all these things happen all the time in a very large number of pathogenic species. And it is very interesting to see that almost all pathogens actually have some basic mechanisms that are common to all of them. It is not that MTB's mechanism is very different from AB. They have their own unique features, there is no doubt about it. But primarily the outcome, efflux or target modification or antibiotic inactivation, those mechanisms remain same. The players change across different pathogenic species, making it extremely difficult to identify a common set of proteins that now you need to work with, right? So we know the complexity of this mechanism. Now, since all of us have been hearing the word pandemic for so many months now, I thought I'll actually bring to your notice that antibiotic resistance is considered to be a pandemic like since 20 years back. So it's just that it's not in mainstream media, or it is not discussed by newspapers. It does not mean it is not a problem. It is actually a pretty big issue. And if you look at this Nature Medicine article from 21 years ago, you can see it talks about the pandemic of antibiotic resistance way back, right? But the world basically woke up to this issue when in 2015, during the World Health Assembly, all the countries, like a very large number of countries in the world came together, and they made a global action plan to address antibiotic resistance. The reason I'm showing you all this background is it will allow you to understand when you do an analysis to understand AMR, what is the kind of impact it will have on our lives, okay? And if you see, there are 5 verticals. We are not discussing 5. We'll be focusing only on vertical 5 and very briefly on vertical 1 towards the end of my talk. And what is vertical 5? So if you really look at how do you address any pathogenic issue or any disease for that matter, there are several ways. Like say, for example, with SARS-CoV-2, all of us have started to looking at those curves, like the flattening of the curve is going up and down, that surveillance. Now that comes from epidemiology. You're trying to see how the virus is going around in the population. Likewise, you need to know how these bacteria move in the population and how do you trace them. Then you can design vaccines the way we are designing vaccines for SARS-CoV-2. I'm pretty sure all of you are vaccinated at least for your first dose. Then there are new drugs. How do you identify new drugs for the pathogenic organisms? And most importantly, and if you see with the pandemic, the first thing that was done was to design the diagnostics. If you can't even know what is there, how will you treat it, right? So diagnostics becomes the key to identifying what are you treating, so that your treatment or your downstream management of infections become possible. And that is why I thought that within this workshop today, we'll focus only on diagnostics from vertical 5. So I'm just trying to drill down to a very focused discussion today. And what happens for diagnostics of bacterial infections in clinic as of now? So as of today, culture, when you say you grow bacteria in a petri dish, remains the gold standard for diagnosis, okay, for infections. Now, what is the problem with that? Why can't we keep using it? The problem is that since culture takes time, the empirical based approach is used for diagnosis and prescription. Again, here, I would like to ask a question. How many of you, when went to a clinician, was prescribed a throat swab or a blood test or any other kind of culture, and after the culture results are received, you were given antibiotics? So how many of you actually got a culture done that was prescribed by the doctor, and how many times the doctor actually prescribed? Even if at all there was a prescription saying, okay, let us see which pathogen is growing, and then I'll prescribe an antibiotic. Has that happened? Yes or no? If it is yes, raise a hand. If it is no, don't raise a hand.
No, it didn't happen. The doctor just note down my symptoms and just prescribe the medicine without any sort of test.
Absolutely. Which means that broad spectrum antibiotics are prescribed to cover as many pathogens, irrespective of what the infection is, right? Now, what that has led to, it actually has led to over treatment, but not the right treatment. So you really are contributing to the emergence of antimicrobial resistance just by not doing targeted therapy. Okay. Second problem is sometimes the biological sample has so low number of pathogens that the sensitivity, even if sometimes you are infected, you don't see that infection in culture. So you don't even know that you are infected. So even if in complex cases like sepsis, you actually don't see infections in immunocompromised patients. So you really don't know whether you give antibiotic or not, because it becomes a pretty life and death scenario when you are dealing with people who are suffering from sepsis. Okay, so and that is why you need to identify a new method. So I have taken an example here. This is a publication I would recommend all of you to read. Now, to put it very simply, what this graph shows you, on the x-axis is the number of hours from the onset of hypotension, which means that sepsis is basically setting in. The infection is generating a very large amount of immune response, and the body is getting overwhelmed with the immune response from the host.
Can you please explain what does sepsis means?
So sepsis basically is that is what I'm trying to tell you. Sepsis is basically the host response to infection. And sometimes the host response is so intense that there actually happens to be multi-organ failure because of the host response itself.
Okay.
Right.
Okay.
So it causes infection, but the damage happens because of the host response. Okay. And what you see on the y-axis is the fraction of total patients who can survive. So as you can very clearly see, the black bar tells you that up to the first hour of onset of hypertension, up till almost 36 hours, which is like approximately 3 days, you have those many number of hours to decide the antibiotic prescription. The earlier the decision is, if you see here, the earlier you actually decide which antibiotics to give, the survival rate is very, very high. The later you decide which antibiotics you give, the survival rate goes down from 80% to practically nil. So it becomes a very, very difficult situation for the doctors and even for the patients to basically take this kind of a decision in such a small span of time, because culture takes time, and then the specificity is issue, the sensitivity of the test is an issue. There are many challenges that the culture is facing, though it actually is helping clinicians a lot. But what we need today is rapid diagnosis. How do we do that? So, and the other problem that I would come back to is, now as you were asking, sepsis is an inflammatory disease. So it's basically the immune response of the host. And if you can see where all sepsis can happen, it can happen practically in all tissue types. So which sample will you take to identify the pathogenic strain? You don't really know, because a lot of body parts are engaged. Basically, they're under the attack of your inflammation. And the pathogen, what do you test for? Would you grow in an E. coli media? Would you grow it in Klebsiella media? Or you an MTB media? You really don't know, because the number of pathogens, suspected pathogens in a septic shock is just amazingly high. And this is only from one study. So you know that when you actually have this huge diversity of infection in terms of the different tissues, in terms of different organisms that can be present, you need something which is more throughput, which can actually give you more specificity, sensitivity, and can work at a throughput level. And that is why you need a method for which you don't have to culture the pathogen. So you don't have actually not taking a pre-decision of what the infection is. It can detect an entire range of pathogens. It should be comprehensive, sensitive, because it should work with very small sample and can detect very minor infections. It should be very specific. You need to know which species, which genus are you talking about. Rapid. Should work with dead and live pathogen, because if you do a culture, if the pathogen has already done the damage and has died, you will not detect it, but it's there. The damage is because of that. And differentiate between pathogen and human cells. Now, this is still a gray area because a lot of human contamination comes when you do metagenomics. So do sequencing-based method offer a help? Yes, they do, because they can identify unculturable pathogens. You don't need to culture them before sequencing. You can distinguish between different pathogen strains. You can identify new drug-resistant determinants, and we'll see that today in our hands-on. And it's an extreme accuracy. So basically, you can actually do better informed treatment decisions. And whatever you get from the metagenomic experiment, you can actually use it to develop specific primers for a PCR-based test, which makes it very, very point of care, easy to handle, because you cannot end up sequencing everything all the time. So if you have something that really works in the population and geographical region that you're working with, an endemic kind of infection, these tests can be really, really helpful. All right. And then what you can do, you have two choices. Either first go a targeted amplicon sequences, which means amplify 1 or 2 or 3 proteins. It is very good because it's cheaper, but it cannot determine new pathogens. It cannot identify new antimicrobial resistance. And obviously it's not comprehensive. The alternative, which is a little expensive right now, but hopefully it will reach clinics very, very soon, is whole genome sequencing, and it can help in detecting novel AMR genes. And that is what we are doing today. In our exercise today, we will learn how to work with whole genome sequence data sets and identify antimicrobial resistant determinants in those genomes. And this is basically for all of you. Point of care means that things have to be very, very easy to work with. This is my Nanopore MinION. It's just like a small pen drive, you know, and then you can load your sample, plug it into your computer, and what you have is the genome sequence of anything that you wish to sequence. So this technology, not just the size of the instrument, but the chemistry behind sequencing has changed the way in which we look at whole genome data sets for a very wide variety of applications, not just bacterial bioinformatics. It has impacted everything that has to do with sequencing. So chemistries like this are very, very interesting to study, and maybe I'm pretty sure some of the earlier speakers might have covered that. So what we are gonna do today is we are going to basically learn to handle whole genome sequence data sets. We'll do pan-genome, and I'll tell you why we need to do that. And then we need to identify antimicrobial resistance genes in these genomes. So if there is an X patient that is not responding to an antibiotic, isolate the patient sample, sequence the bacteria, and you know why, you know, a priori, without even giving the antibiotic, you can actually say this patient is not going to respond to A, B, C antibiotics because of the molecular signatures that I can see in the protein, in the genome sequence. Like say, for example, it has a mutation at certain position, which is leading to target modification, or it has basically acquired a new gene that is responsible for antibiotic inactivation. Or I could also see, which I cannot do with genome sequencing, obviously, but they have to do transcriptomics for that to see the efflux pump gets overexpressed and a lot of the drug is being pumped out. But a lot of this can be done with omics technologies, okay? Now, before we make an assumption that if we are talking about, say, for example, E. coli, are all E. coli same? They are not. Are all human beings same? No, we are not. We are very different in the way we look, we talk, we think, our predisposition to infection, right? We are all Homo sapiens, but we are not the same thing, right? The understanding is because we cannot see bacteria and they all look like small colonies on petri dishes. Assumption is they are all the same is also not true. They're actually very different. And that is why you need to do a pan-genome analysis and not just work with one sequence, because it's going to give you a very limited understanding of what that species can actually do, okay? And one of the first papers that actually identified this was this paper that came in PNAS almost 15 years ago. And what it says that strains of a bacterial species might differ substantially in their gene content, okay? Which means that they are not the same. And total gene pool of a species might be orders of magnitudes larger than the gene content of any single strain, which means that the number of genes that the entire species will have is way beyond the number of species, number of genes any of the isolates will have. So looking at one isolate will not help. You need to look at the entire repertoire. Now, how do you know how many? Is looking at 100 E. coli enough? Is looking at 1,000 E. coli enough? Is looking at 10,000 E. coli enough? How do I know when to stop? We don't know that, right? We cannot arbitrarily decide, okay, 50 is a good number. In order to do that, what a lot of researchers have done over years is to develop something which is called as a pan-genome analysis. And there are like several pipelines available. I have just picked one of them today to basically make you understand how a pan-genome works, what is that you need to look there when you have and done the analysis. So just to proceed forward, so say for example, I have taken 3 genomes of E. coli, 3 clinical isolates of E. coli. I sequence them and then I do pan-genome. What does pan-genome tell me? It tells me first, what are the set of genes that all the 3 E. coli strains share, which means it is present in all the species, all the strains that you are looking at in that particular experiment. Okay, now that becomes the core, as in core, present in every isolate that you sequence. Then what you have are dispensable, also called as accessory genes. They are present in 2, 1, maybe 2 of 3. So those are dispensable. But if there are certain genes present only in one of them, as in it's not shared with any other strain, then it is a unique gene. And this happens. And as I was telling you earlier, how will you decide when to stop? Okay, like say for example, then I basically plot a graph. What I do on the x-axis, I will mark the number of strains I have sequenced. First strain sequence, second, 3, 4, 5, 6, 100. Okay, and how many genes is each one of these strain is contributing. So when I say total set, I mean what are the new genes, right? So imagine if my pan-genome is closed, which means that irrespective of how many genomes I add, my gene number is not going up, which means that I have captured the entire diversity of that species, and it is okay to convincingly say it's a closed pan-genome. 120 E. coli are enough to close the pan-genome. There is nothing that you can add. So even if I sequence 100 more E. coli, I am not adding to the genetic diversity of the E. coli species as such. On the other hand, if my graph is like this, as in if it is still open, which means every time I add a new genome, the number of genes keep on increasing, which means that I have not yet covered all the diversity. I need to keep sequencing more and more isolates till the time this line becomes a flat. So that addition of new genomes is not adding new genes. That is when you can call it as a closed pan-genome, and this should be your target, because then you know you are talking about all the diversity possible. And again, this is a graph which shows you that the size of the pan-genome increases, but the size of the core obviously will decrease because the core has to be shared by all the species. So more the number of species you sequence, the chances that the core will reduce is logically going to go down. Okay. Now, why this is important? So the core genes actually has been observed and are responsible for basic aspects of the biology of the species and its major phenotypic traits, because obviously all of us are human beings. So we are similar in most ways. We both have 2 eyes, we both have 1 nose, 2 ears, right? So the morphological details are same because we actually have the same structure. So the genes responsible for that structure are the same, but that's how that structure will look. That is where you see variation, right? Likewise, the accessory genes, which is not shared by all the genomes, by all the strains in a species, are basically supplementary biochemical pathways. Now they give an extra edge. Like say, for example, some of us are pretty tall. Some of us are very good athletes. Not everybody is like that, right? Now those are additional traits which offer selective advantage, like such as ecological adaptation, virulence mechanism, and even antimicrobial resistance to these species. And that is why they have it, right? And so the point is, general assumption would be that initially if a bacteria is evolving, you would see these antibiotic resistance genes indispensable. Some will have, some will not have. But if that mutation or addition of that gene gives the bacteria a survival advantage, makes it more fit for transmission, then it will get fixed in the population and become part of the core genome. And that is exactly what we want to know. What has reached dispensable, what has reached core, and that will allow us to understand how many times, like what kind of outcome we can give when a clinical sample comes for analysis. So for today's demo, and it's a demonstration, so I would recommend to you to listen to what I'm trying to say rather than do a hands-on parallelly. The reason is some of the steps in the demo are going to take a pretty long time. So it will be difficult to do all the steps as I am doing the demo, but re-watch this video. Wherever there is a step that is taking time, stop the video, finish the step and go to the next step, right? So for today's demo, I'm working with this paper because
Excuse me, ma'am.
Yes.
Can I have a question?
Yeah, please go ahead.
Okay, ma'am. It is an excellent talk. Just I have one question here to your last slide. So actually this pangenomics concept is across species. So you have told about different strains of E. coli. So in case of microorganisms, is it like that? It is across isolates or strain because it is the same species. What I understand.
So yeah, you have raised a very valid question. The answer to that is at what resolution you want to do pangenome analysis, it depends on you. If I am interested in doing pangenomes of all E. coli strains, then I can do that. I can say I am doing it only for Escherichia, or I can actually go in taxonomy further up and say I am doing a pangenome for all gram positive and all gram negative. So it really depends, because you are trying to apply a method in which you are trying to map what is the gene content that these subset of organisms share. Okay, so you can do pangenome at any level. Now, answering your question, are there strain specific differences? Yes, there are. And most of these differences have been observed in antimicrobial resistant strains. So they are not all the same. There are genome level differences. And in some cases, like in case of non-tubercular mycobacteria, which is a completely different story, you see that they do a very, very, they actually share a very large chunk of their genome for horizontal gene transfer with other NTMs, with Pseudomonas. So yeah, it's a whole new world out there. If you see how bacteria intermingle with each other and they keep sharing the genetic content, it's just amazing. And Mycobacterium tuberculosis, the reason for taking this example for today's activity was that in most cases, MTB actually shows drug resistance via mutation and not so much via horizontal gene transfer. Okay, and moreover, it does not have plasmids, so it makes your life simple. You are only looking at the chromosomal DNA and not worrying about everything else. And the second reason is this paper actually has three things. It talks about machine learning, it talks about pangenome analysis, and it talks about AMR. So I think it's a very good exercise paper for all of you to get hands-on and really learn what goes into understanding the AMR content of any genome using a lot of these online tools that are available. So it's a good exercise, okay? And for this, what I would recommend you to do is when you actually go to this paper and you scroll down. So have I answered your question?
Yes, ma'am, it makes sense. Thank you.
Makes sense, okay. And I can see there is a question posted by Sharbana. It is saying that, is it possible that unique gene may be integrated in the core genome in case of MDR strain? It can, but before getting widely, you should see indispensable first, right? How from one strain, it'll suddenly go to every other strain? That is not feasible, right? So there's an evolution. Even if a new gene is acquired, it will then circulate. You will see, okay, when I have sampled 100 people, and I can see it in one. But over a period of time, I can see it in 50. But over a period of even more time, I can see it in all 100. Like the way you see SARS-CoV-2 strains right now, right? They are mutants. But you don't see one mutant just infecting everybody on Earth at the same time. You see circulating there, these mutants are basically increasing in number over time, right? So this is what happens. It's an evolutionary process to some extent. So a unique becoming a core directly seems very unlikely. A unique may become actually accessory and then core very, very quickly. So yeah, it also depends on what you are sampling, and it also depends on at what time duration you are sampling. So yeah, so that is what I understand when I say pan-genome analysis and fixing of different mutations and genes in a population that is making the bacteria more transmittable, as in giving its superpower to not just avoid antibiotics, but also becoming more transmissible. Now you have to remember one thing in mind. The point is that if this was the protein that was working in very happily, functioning in a bacteria, right? And what you did, you actually generated something which is a drug, and this drug comes and binds here, right? And now what has happened is this function of this protein going this is not happening, right? Now, which means that when this function stops, the bacteria dies. Imagine the critical nature of this function. And now imagine that bacteria is trying to mutate this protein. When this was blocked, the bacteria was not able to survive. Now it has left with the challenge to mutate it in some fashion that this is not fitting in, but this is still working, right? Which means initially it is compromising its fitness, and then via compensatory mutations, it is regaining its strength again. So you need to think in a very logical fashion that the bacteria is trying to evolve a particular protein in its physiology, which was very critical, and it's not easy, but still it's doing that, and it's doing that pretty successfully. So that is something at a molecular level we need to understand better so that we can address this issue better. Okay, now let's come on to the demo. So if you see this paper and you scroll down to the end of the paper, there is a section which is called as code availability. So what they have done is they have provided their entire code and data on GitHub. Go to GitHub, and when you go there, the GitHub, you click on data, because we are not really running the Kavas pipeline. We are actually doing the other pipeline, which is simpler to understand for a demonstration. So go to data. And when you click on data, what you see is a very large number of files. What we are interested in among all these data sets are strain information.csv. Okay. So basically what we are trying to do here, we will take a set of MTB clinical isolates. Their whole genome sequence is available. We are going to take a sample of that, do a pan-genome analysis to see how the core looks like, how the accessory looks like, okay? And then also use these sequences to identify antimicrobial resistance genes. So it's a very simple exercise, but this is how you will be, it will be simpler for you to do it, okay? So when you click on strain information, it will not let you download that CSV file easily. So you need to go there and then say download. Once you download the file, how does it look like? The file looks something like this. What it has a very large number of columns, and you have the genome ID. Then you have this genome ID belongs to which strain in MTB. Then you have the organism, then taxonomy ID, then a lot of other information related to each clinical isolate. Okay. What we are going to do is, because I don't want you to take a very large data set, this paper I think has almost 1500 plus clinical isolates. I'll be taking only 5 or 6 today for our demonstration, just to show you how the pipeline operates. Now, if you see here, this data is actually coming from Patrick. Okay, I don't know if anybody covered Patrick before me, but I just want to show you what it is. So Patrick is an online system where you can actually either have a very large number of genome sequence data sets along with their AMR phenotypes available. So I would recommend that you may start using this resource. It's pretty easy to use. And if you see here, the genome IDs present in the paper here are exactly the same that Patrick gives, because even in the paper, the authors have used data from the Patrick repository. Now, point is, for today's demonstration, we are using 5 or 6 sequences. I can obviously say, okay, click, 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 and download. What if you need to do it on 1000+ sequences? You cannot keep doing that, right? So you need to, to some extent, automate this process. And what I have done for today's demonstration is I have kept the analysis part in a manner that it is semi-automated. It is not completely automated, because I can understand from a lot of WhatsApp discussion is that a lot of non-programmers are there in the community. So if I really don't want you to learn coding and then do it, you can practically do today's demonstration hands-on without doing any coding. So yeah, you can actually finish the entire pipeline of pangenome as well as AMR mapping, and you don't have to write even a single program. Okay, so that is what we are intent to do today. So now if you can see, we are interested in 5 of these. Now, how do I automate the download of, say, for example, 5 or 1,000 such genomes is, I'll first of all, I need to convert my genome IDs correctly. Now, this is where the detail is important. See how the genome ID Patrick is giving? It is giving a number, then there is a dot, and there is a version number. But if you look in the paper, they have replaced the dot with an underscore sign. I really don't know why they did that. But if you ask Patrick 1010,834 underscore 3, it will not know what it is. You need to ask 1010,834.3, because that is what it recognizes in a search. Okay. So first things first, we need to convert these underscores into dots. All right. And then the second thing is, once we have done the conversion, so we have actually can use Notepad in one go, say replace all of your underscores with dots, and that is where you have. So I've selected these 5 genome IDs, and now you have genome IDs that are recognizable by Patrick. Now, how will I, without going to Patrick, can download the genome sequence datasets? It's pretty simple. Whether you are using a Windows machine or a Linux machine or a Mac machine, there is a utility which is called as Patrick command line. I would request all of you to install it. And all the tutorial that I'm showing you practically should work on all the operating systems except for the last step, but it'll work on all of OS. So when you actually have Patrick command line installed, and this is a screenshot from a Windows 10 system, just to make it easier for you to see how it will look like. And I open this, what I need to then do is I need to type a command for this program to take my genome ID, go to Patrick, fetch the genome sequence data, and bring it back to me. Simple step. Okay. So how do you install Patrick command line is all given here. So I would recommend go here, and there are a lot of, so you need to ask a question, and Patrick allows you to download data in like 1,000 different formats. So it makes life very, very simple, believe me. Learning Patrick CLI will solve a lot of your day-to-day data handling problems in a very simple fashion. And they have a lot of scripts that they call as P3 scripts for that. Okay. Now, how these scripts work, we actually are interested in one of the P3 script, which is called P3 Genome FASTA, because we are interested in providing the genome ID that we picked from the paper publication and getting the genome sequence data for us. Okay. So this is a very simple command that we are going to use, and see how easy it is to run it. Now, this is what we have, right? 1, 2, 3, 4, 5 genome IDs. How do we get the sequence? When you open Patrick command line, just say P3 genome FASTA, give the genome ID a greater than sign and any name you wish to write. I just followed the same genome ID so that I can follow up. You know, I know that this genome, this particular FASTA file belongs to this particular genome ID. You just have to do this and do this 5 times. Now, this is a step for programmers who can really write a simple for loop and can just run it. People who do shell programming can also automate this step for more than 5, 6, 20 sequences. Till the time you are not dealing with like 50 plus, it's still manually achievable. Yeah, 50 beyond, I would not do that, because, and you know, it's not about taking time. You don't want to introduce errors while doing git analysis. So to avoid errors, automate it after a certain point. Don't do it manually, because manual errors are extremely impossible to trace. Okay. So now what we had these 5 genome IDs. We said P3 genome FASTA, gave the genome ID, put a greater than sign, and stored this genome ID into a FASTA file. What I'm talking about, where is this file? This file goes in here. So you have all these sequences available. And when I click on one of them, this is how it looks like. So it is a single chromosome. It is the whole genome sequence of 10108834_3 FASTA. This is the whole genome sequence of a clinical isolate that I was interested in doing pangenome for. And likewise, I have now 5 such genomes available with me. Okay. What I'll do next, now I have these 5 files. Each file is a whole genome sequence of 5 clinical isolates of MTB. So far, so good. Okay. What next? Now, what I'll do is I'll go to Galaxy, and I'm pretty sure Kiran and Sangram are giving you a pretty decent tutorial. I have attended a couple of them and they are very good at it. So now what we'll do, we'll go to Galaxy and we will say upload. We all know how to upload data to Galaxy, and we select all these 5 files and then say start, and it will start uploading. And then here it is. So what it has done, it has uploaded all your files and placed them in your history. I am assuming that you know Galaxy, and that is why I am not going through the details of the Galaxy interface. All right. Now, once you do that, what you will then have to do, you have to run something which is called as a PROCA tool. Okay, I'll come to here first and then go back. So there is a Galaxy training tutorial available on genome annotation with PROCA. Now, PROCA is something that takes your genome sequence and annotate it. What do I mean by annotation? Is I'm basically adding punctuation marks to the genome. What does that mean? Imagine there is a horror novel or a thriller novel that you are reading. What I do is I remove the table of contents. I remove all full stops, spaces, and commas everywhere they are. I remove the paragraph differentiation, chapter headings, nothing, no punctuation. Everything is written as a series of alphabets. Can you make sense out of it? You cannot. After a point, you won't be able to, you can say, okay, and I don't know what it is, and you don't know where to stop. So that particular string of characters will stop making sense if it is a contiguous stretch of characters without punctuations. When you sequence a DNA, like when we get a DNA sequence, it is exactly like that. It's a stretch of a sequence. What does it mean? We don't know that. So tools like PROCA allow us to basically add punctuation marks. It will say, okay, from position X to position Y, there is one gene that is responsible to function as X protein. Like say, for example, DNA polymerase, RNA polymerase. It is doing something. And now you actually get to add meaning to the sequence data. And that is what we need to do first. We need to first do a structural annotation, identify from what point to what point. It's one word, it's one sentence, it's one paragraph, it's one chapter, right? So the annotation will also depend on what you are trying to read. You're trying to read one word, a sentence, a paragraph, a full chapter, the whole novel, right? So it depends on what you're trying to read. And PROCA allows you to define that structure in place. There are many tools, but the reason we are using PROCA today is because the pangenome tool we are using today, Rory, works only with PROCA output files. It doesn't work with any other things. So when you do say PROCA, and Galaxy already has PROCA and Rory both. So you say, you basically upload your sequences, go down, scroll down, and just look for GFF3 format. You don't really need all of this if you're running Rory. It only looks for the GFF files, okay? And that is when you say, okay, execute. The minute you say these are the 5 genome sequences we gave, the minute you say PROCA is running, we all know that Galaxy starts to show it on your history that, okay, it is generating your GFF files, all right? And after a while, it becomes orange, and after a while, it becomes green, which means your job is done. Now, I just click on one of the GFF file for one of the genomes to see how it looks like, okay? It's a very simple file. It's basically a file with several columns, and it is important that each column has information on the annotation, right? And normally GFF files came into existence because we want to basically visualize these structure, this genetic structure on the chromosome of any species, right? So say, for example, the first column here is your genome ID or your sample accession number. Then you have a prodigal ID on the basis of which PROCA does annotation, whether it's a CDS or a tRNA, it will tell you what type of annotation it is. And then the most important thing is, what are the coordinates? From what position to what position are we talking about this particular? Say, for example, where is DNA A? So DNA A is from 34 to 1524, and it is on the plus strand. Likewise, on this position, your this particular, say, for example, let's take another gene, which is on the negative strand, CRGA. CRGA is from this position to this position, but on the other strand. So this is very critical information. And what PROCA has done, it has actually provided you gene sequences and as well as protein sequences of this particular data sets. What Rory reads is these GFF files. And then based on this, it will try and compare what are the common genes across all the 5 pathogens and what are unique and what are accessible. It's as simple as that. Okay. So Rory is available from here. It's a pan-genome pipeline. I like it because it actually provides output in a very simple to understand fashion. And if you scroll this page for Rory, you will actually find a very large number of programs that will take the output of Rory and give you many other things. So that is why I recommend this. And then unfortunately, when I was trying to run Rory on Galaxy, it was not running. And when I went to the Rory platform, they actually tells me it's a bug. So that once they resolve the bug, Rory should be working on Galaxy. So don't lose hope. I think it's a matter of time that Rory works on online, so you don't have to struggle with installing it. I installed it, and it's a very simple input. I'm saying create a folder pan-genome, and -R is basically for Rory to know that I need to see plots. I need to see graphs. If you don't say -R, it will not plot anything. It will show you a lot of files, and I'll show you what kind of files it tells you. And then all the GFF files, right? Now I've just written star. I could have, mentioned by name, but you need to basically take this is the most comfortable input. You just say all GFF file in this folder, and this this folder has 5 GFF files from the 5 clinical isolates we used. All right. And this is the output of Rory when it has finished execution. It will give you a very large number of files. What we are interested is on primarily a few things here. I'll show you which ones. First, this is the graph. So since we used 5 clinical isolates, it is showing me 5 genomes and number of genes. Now what it is showing me, that the conserved genes are going down as I added more genomes, but the total number of genes are going up. Now it clearly tells me that 5 genomes are not enough for MDB because my pan-genome graph is going up. We remember that from the previous set of slides, right? So if it is not a straight line and if it is going up, it means I need to sequence more. So in very quick analysis, 5 is not enough because my total number of genes are still going higher. The other thing it tells you that when I add new genomes, are there any new genes that get added? So you will see that, of course, with the first genome, you added all the genes, but at the second genome, you actually haven't added much. Most of it is that they share. And as and when I'm adding more genomes, new genes are not really adding, but my unique genes are going up. Because as and when I'll add more clinical isolates or more genomes, their uniqueness will also get reflected in the data set. And this is what you will see here. Okay. The other thing that you see is summary statistics. Now this tells you the what we discussed. So core genes, we know it's 99%, basically between 99 to 100% strains. If a particular gene is shared, it is called as a core. So when among these 5 genomes, almost 3,712 genes were shared among all the 5. So that becomes core. There is a concept of soft core, in the sense, don't be too stringent. Just see what happens between 95 and 98. So I couldn't find anything in this data set, but I'm pretty sure if you increase the data size, you will see that. Shell genes are something which is 15 to 95%. Now this is nothing but your accessory genome, shared among some, but not all. And this is a pretty huge number. Then cloud genes, again, it is basically telling you about the unique genes present only in one and not the others. And this is a total pan-genome, which is 4,600. Okay. And one file that comes as an output of Rory, which is very important and very important for you to know how to read it, is this. What it has a list of genes, which is nothing but these 4,600 genes that you see here. Okay. And then the second thing is their annotation. The third is the number of isolates in which these genes are present. So if I need to know this list of 3712, how do I know which are these 3712? I simply have to go to this gene presence absence.csv file. If it is present in all 5 isolates, I know it is core. If it is present in 4, 3, 2, I know it is accessory. If it is present in only 1, I know it is unique. So just by looking at column C, I can make a lot of estimations, and I know exactly what I'm talking about. So which are these ones which are core? What are the ones which are accessory? Which are unique, and in which strain? So you know if it is unique, this is unique, but it is unique only in one and this one. And then I can go back and check, is there a specific antibiotic to which this strain was not responding to? And if it has this unique gene, maybe I have discovered a new mechanism of resistance, right? So that is how this kind of data allows you to explore and identify new things which are not possible if I was sequencing only one. But since now I have this data, I also know that these particular strains are responding or susceptible to certain antibiotics. This data allows me to evaluate now the phenotypic association. Okay. Now, just finishing the last few slides, how do you know which of these genomes already has an identified antimicrobial resistance gene? How can you say it is new? You don't even know what is already there. So what you do is, there are again tools available in Galaxy. Abricate is there, Star AMR is there, there are others also. I have used Star AMR. So what you need to do is, we already imported all the genome sequences file in Galaxy. Select on any one of those and say, okay, execute Star AMR. When it executes Star AMR, it actually gives you a very large number of files. Primarily, our interest is to understand what is the data that is giving from ResFinder, because it tells me what already AMR genes are there. Now, in this particular strain, it is showing that the predicted phenotype is that this strain should be resistant to gentamicin, and it should be resistant to some macrolide. We don't know which one, but because ERM is a gene that is responsible for macrolide antibiotic resistance, so you know that this has at least has 2 genes that are already responsible for 2 different classes of antibiotic resistance. Okay. Now, this is not the only resource that one should use. There are Abricate, there is CARD, there is TRAP. There are many analysis pipelines. Use all of them because antimicrobial resistance is a very complex phenomenon, and not anybody has able to map everything. So you will find a lot of new information if you start exploring as many number of resources as possible. And based on comparison, now if I go back and check whether AAC and ERM are in core, are they an accessory, are they unique, it will give me an understanding of how critical these genes are in my data set, right? If it is in core, I know that at least in this particular section, geographical region where I am looking for these samples, AAC mutation actually is responsible for gentamicin resistance. So if I have seen sequence data, I will not prescribe this individual gentamicin because I know it's not going to work, right? And there again, antibiotic resistant detection is also available in Galaxy tutorials. I have not covered any of the plasmid associated resistance today because that is again a different story, but this tutorial allows you to explore that vertical. It actually tells you how to assemble genome from a nanopore data, and it's a stepwise process. I would recommend take a look at it. It's pretty simple to understand what they're doing, but do it. And that is the only way to learn. And the last point is that some of these sequencing platforms like Nanopore already come bundled with these methods like WEMP, which what's in my pot kind of situation. So that is where you need to, these are basically tools that when you sequence, the minute your sequencing data comes out, it actually comes out with a very large number of annotations already done. So you don't have to do them, but you of course can do further annotation. Now, tell me one thing. All right. So I can see Prashant has left. Other people are around. And how tired are you? I'm gonna take See, at least 5 minutes more.
Can you hear me?
Yes, Kiran?
Yeah, actually there was some problem in the host. So right now Anuba is host. So my PC got hung up, but I hope the recording is happening, but still, yeah, Anuba is
No
host.
problem. No, I was saying I can take 5-6 minutes more, right? Because that is all that remains.
Yeah, please go on. Please go on.
All right. Okay. So the thing is, now coming to the last thing that I mentioned in the Excel sheet is, a lot of machine learning based methods are now applied to understand which genes are antimicrobial resistance. And why do you need to do that? If you just focus for next 1 minute on this slide, what I'm showing here that a person with at the time of diagnosis, which is 0th month, comes with a sensitive infection, as in it was treatable by antibiotics. And this is a classical case for, I'm showing it for tuberculosis, right? And at that time, if you sequence it, you know that the sequence of a susceptible organism looks like this, okay? Then after a few months, you see that the person has developed resistance to these antibiotics. You again do sequencing and you again have your sequence data. Over time, the same set of patients actually develop MDR, XDR infections. So they go from sensitive to MDR to XDR. Like they're not even responding to a very large repertoire of drugs now. You again sequence them. Now, machine learning method allows you to identify which genetic components can segregate susceptible from resistant infections. And once you can do that, it allows you to, from a mixture of clinical samples, do a pretty decent estimate of the drug resistant status of that individual. And it will also allow you to discover new drug resistant determinants. And this is what is a very, very hot topic. There are a very large number of tools. This is one of them. I really like it. It's a K-MER based strategy. I'm not describing it today, but these are the applications that are available. The other thing is, do you really think it can ever reach clinic? It actually has already in some places. So in 2018, the New England Journal of Medicine actually published a paper that how prediction of susceptibility to first-line TB drugs is already happening by a DNA sequencing. So you are not now supposed to do microbiology. You don't have to take sample, grow it, and then decide, okay, isoniazid is working or not, if amplicon is working or not. No, you can actually sequence it and say, I have this gene, I have this mutation, it will not, the patient will not respond to isoniazid. So that kind of one-to-one correlation is already started happening, at least for TB, for first-line TB drugs. So it is not something which is futuristic, it is actually happening right now. And given that we all are going to, basically we are already getting our RT-PCR done, right? And everybody is doing it for SARS-CoV-2 infection, and the labs or the hospitals are giving you reports. These are all molecular markers. I think it is high time that even as society, even not as researchers, we need to understand when they give us that data set, what does that really mean, right? And this is the one thing that I would really like to very briefly cover. The whole point of doing sequencing was that we should not be going through the step of culturing, right? And that is when metagenomics come into picture. You take the entire patient sample, sequence it. And once you have sequenced it, you basically then identify what strains are there, what species are there, what is their drug resistance profile. And Galaxy actually gives you a very large number of tools to do metagenomics. And once you have identified what bacteria you're dealing with, you can then do pangenome, you can do AMR mapping, right? So that is the pipeline that you should be thinking about. And these are basically certain terms for you to search literature. This is not basically, you already know this, but this is something that will allow you to look for the right papers. Like the terms like clinical metagenomics. It refers to the same concept that you take DNA from a clinical sample and recover as much information as possible. And traditionally it has been called as salvage microbiology. So you basically are trying to detect bacterial DNA directly from clinical samples without doing any culturing, right? And they allow you to detect pathogens. They allow you to make estimates of which antibiotics may not work. And a full drug susceptibility profile would definitely enable directed treatment and not the way we are treating right now, which is actually causing two problems, AMR as well as exposure to ineffective toxic drugs. Your system will also respond to something which is not good, right? Okay, so what we have learned today, there is a lot of data and there are a lot of priority pathogens to be addressed, at least 12 of them. Now, can we apply feature selection machine learning based methods to basically identify which, what are basically genomic variants or genes that are responsible for AMR to different antibiotics? So that is the take home message, that how do you convert data into knowledge? You have a lot of data and today we converted it into knowledge to some extent. We took genome sequence data, did the annotation, did the pan genome, concluded certain things, mapped it to AMR phenotypes, we identified certain new things. So this is something that is a very critical aspect of how do you understand phenotypes. And in this case, phenotype is drug resistance from genome sequence data sets. Okay. Now, towards the end of the talk, I would really like to tell you that correlation is not causation. If a statistical method is showing you something, have your doubts, okay, and do your homework and try to make sense out of it the way you understand the biology of the organism, okay. I'll give you two examples here. Now, the first graph is a graph which is it's a very strong correlation between these two lines. What are these two lines? These are two lines between the number of people who are drowned by falling into a pool versus films Nicolas Cage appeared in. Logically, it makes no sense, but there is a very strong correlation. So when something is defined your knowledge, or when something is defined logic, don't follow it. It can be a very strong statistical correlation, but it can mean nothing. Second, US spending on science, space, and technology correlates with suicides by hanging, strangulation, and suffocation. Man, look at the correlation. I really don't know if it has any meaning. So just don't go by looking at graphs. You need to apply your knowledge base when you do this assessment of the data, right? So AI can be there, computing can be there, but nothing makes more sense than common sense, okay? So apply that all the time when you're doing science, because running tools is one thing, but what are you extracting out of it is altogether a different thing, and that is why you need to read as much as possible, okay? Read about what you are dealing with. And let me see if there are any questions. Okay, now coming to the last part, and this is basically not to teach you anything. I actually need your help. And this is why I thought I'll use last 1 minute of my presentation to just seek for your help. Now, I told you that we will be focusing on two verticals today, 1 and 5. 5 we practically touched upon in some fashion. 1 is to improve awareness and understanding of antimicrobial resistance. How do we convey these very difficult to understand concepts to wider community? And I thought, why not start with kids? Because if kids understand something, it will be easier for them to explain it to others. So I basically ventured into something very, very unconventional for a research laboratory. I actually ventured into developing scientific video games, and Armor is my first contribution to that. So Armor is the game on, is how the landing page looks like. And it is to basically trigger scientific curiosity and awareness. And the whole point is, it actually conveys you while you are playing the game that antibiotic misuse should be avoided, less or more. Either way, it is dangerous. The game is already endorsed by WHO. So yeah, they feel that the game has value. And right now it has 6 game levels where we are talking about skin infections, 1 game level on lungs, and 1 on COVID-19. Okay. And this game was covered a couple of months ago by Times of India. It is currently supported by a very large number of funding agencies, and currently Jigyasa is heavily supporting the development of the game. And the point is, as I told you, it has 6 levels. And the overall message is, don't over or underdose. The minute you do that, you are not going to go to the next level. This is how it looks like. And these are the URLs where you can read more about the game. But primarily, the reason why I'm talking to you about it, I want beta testers. I want really you to play this game and tell me that it is crap, it's not good enough, or it's good, but it needs improvement, or it makes sense and you would like to add more levels. You know, it can be any kind of a recommendation, it can be criticism, it can be a suggestion. And primarily it has a lung world and a COVID-19 level were added because we wanted to show that some treatments need combination therapy, And also the fact that everybody is talking about COVID-19, but people know that TB is a much bigger problem, and somehow it gets shadowed by what's going on right now. And that is why we wanted to bring it to the front. So it's important that we do that. And COVID-19, of course, I don't need to tell you why it is there. And the second thing is, this is basically an invitation to beta testers. And what are beta testers? You are real users. So you play the game and say, I learned, or I enjoyed, or I don't like it. There is a form that you can fill in terms of giving recommendations of what we can add. And this is the registration for beta testing. Maybe I can add this link to the chat window once the talk is over, which it is almost done. And I would like to basically take this opportunity to thank a very, very large number of students who I've worked with. It's because they taught me a lot of things. Believe me, I have learned more from them than I actually have ever given away to them. And we actually published a lot of papers together. These two are my favorite. I think all the other students may get really annoyed, but these are the youngest guys I work with. So Pooja came to my lab when she was in her 12th standard, and she actually published a Nature Scientific Reports paper as a school kid. She is the joint first author with Arun. And right now, and then after that, she became the Gates Cambridge Fellow, and right now she's doing her PhD in cryo-electron microscopy from York University in the UK. She's amazing, but then she worked pretty early on in her career to deliver something which was astonishing. And if you talk to her, you know what I'm talking about. She knows what she is doing, and she reads a lot. The other most recent good news I heard is from Ashma. She came to my lab for a couple of weeks to basically try to understand what she will want to do. And in the middle of that, we end up writing a book chapter together. And then she identified that this research is something that she wants to do. And she was just in her 11th standard. So she's probably the youngest kid I have worked with. And now she went on to become the Goldwater Scholar, which is a prestigious fellowship in US. And she also got a Johns Hopkins University admission with a full scholarship. Now, this talks of how these young kids are really making strides. And I'm very happy that they acknowledged they're working with my lab so that they could do this. But believe me, these guys are already very good. It's just that, you need to believe in that they can do it. And there is no limit to what these people can achieve. And I like this. I really like working with a very large number of undergrad students. These are from Delhi University. This is from Miranda House, Shastra University, and this is at CSIR 4PI. So, a lot of people at a lot of places. So I have a pretty large student network, and they really teach me a lot, and we have fun working together. And just to conclude, these are all the projects that I have did over years. It is for any potential PhD students out there who would like to join my lab. My focus of my work is genotype-phenotype correlations and AMR. So if you're interested in these kind of projects, this type of science, please write to me. And I have, this is my lab here in India. Anurag has already left for UCSD for a postdoc. Dhruv and Kishan have left for IBAB. And there are a lot of other students who are doing pretty amazing work. And this is my lab in Paris. Rania and Rafael were my postdocs. And then these two students were working with me on a summer project. So yeah, I had a pretty good experience leading labs in India and France, and it gave me a pretty decent exposure. And I had a very, very good community there to work with. I have my email address here. I have my Skype and my Twitter here. Feel free to connect me through any channel. I am practically available online all the time. And just to sell my institute a little bit, when you look at, there was a research done by Database Commons. And if you see, Institute of Microbial Technology has contributed some of the top ranking databases in the world. And we are in line with EBI, NCBI, and other places, So this place is very good for doing in silico data analysis work. So do consider this as a place for your future PhD. Our campus also looks pretty fancy. So, and I'm looking for passionate PhD students. So if you are looking for doing your PhD, I'd be very happy to talk to you. And with that, I would really like to thank. And I think I have taken a little bit of extra time than was allotted to me. I hope it's okay, and I'm really not keeping you out for dinner. So yeah, that's pretty much from my side. And if there are questions, I'll be very happy to answer them.
Ma'am, can you
Excuse me,
share
ma'am.
the links in chat box? The
What is
game.
it?
Hello.
Yeah.
Armor game. Hello.
Yes, I'm doing that right away.
Okay, ma'am. Thank you. Okay, ma'am. Thank you. And it was a very nice talk.
Oh, thank you.
Excuse me, ma'am.
Yes.
A very, very inspiring talk, of course. And the question is, it's a general question. So regarding this statistically significant. So how to deal with this? Because very often we face, even I face, so even I don't agree with my supervisor. Even I don't find a way often. So actually how to deal with it? Could you use some limelight? Could you give?
I understand what you're saying. So my solution to that problem is to look at multiple evidences. If there is a single evidence pointing to a particular fact, I would try and do something completely complementary to it and see if it really fits in. Of course, there will be outliers in your data that will not fit anywhere. But if your major hypothesis is supported by more than one approaches, you can believe it, but never by one. Try at least 2 or 3 different ways of looking at the data, keeping the question that you have in mind. Generate evidence to believe that your hypothesis is true or what you're trying to see makes sense, and use more than one methods to look at the data sets. Not rely on one ever. Like I never do that because it can be extremely difficult to convince the reviewers. It'll be extremely difficult for you to convince yourself. And then what you're telling to the readers is some kind of a misguiding them, like oversimplifying a very complex system. So let's not do that. Claim as much as we see. You don't have to solve everything. So just basically claim as much as you see there. And most of the time we go overboard and claim bigger things than the data is trying to tell us. So just stay very, very close to what the statistical method was supposed to tell you. Interpret as much and have another independent method to verify what you're trying to say. That normally works. If you are really interested in making sure that your observations go to the next level.
Okay. I suppose that was the last question, I suppose, or do you still have any questions, others? If you have, you can please go on. Because anyway, I think the next session, I think Prashant wanted to take, that is now, it's not dropped. So that will not happen. So this is the last session. So if you have more questions, you can ask.
Ma'am, can you please show us live demo of how to download that CSV file? Because when I am clicking on it, it is opening it another tab, or I can present my
No,
screen.
I understand what you're saying. But just do one thing. Anywhere on the page, do a right click.
Right click, okay.
And then save link as.
Save link as, but it is showing that strain information, it is showing save link as HTML.
No, it won't. Can you just show me your screen? I'll tell you.
Yes.
Yeah, let's resolve it right away.
Can you see it, ma'am?
Yes, so click on strain information.
Left click.
No, yeah, the regular click.
Okay.
Go to download.
It is
Yeah,
showing
on
like,
anywhere on this page, do a right click.
okay.
And say save as.
Okay.
That's it.
Okay. Thank you, ma'am.
No, the reason I know this is I tried downloading it the first time I faced a similar issue. So
You can
okay.
yeah,
Okay.
On the GitHub, always there is another option, which is like raw data. That is the option usually you can use. That's also you can do.
yeah, but even with raw data, this data size was bigger now, so it was not loading it. So
exactly, I was about to say that. That will be a constraint. This is the best option when you have CSV.
Right.
Yeah. Thank you so much.
Oh, most welcome. Good. So if there are no more questions, I think we should let everybody have dinner.
Yes.
Please be in touch, and I really look forward to receiving your registration so that I can send you the link to the game. I would really like you to play the game and criticize it as much as possible so that I can, we can actually make improvements.
Okay. So that's all, I think so. Yeah. So thank you very much, Anshu. Really, it was really very good. I mean, awesome talk with the basic to kind of going to advanced also, I would say, a bit of advanced to students. And of course, and as usual, you were really awesome in giving the motivation kind of a thing, other aspects of the things. I really remember that we met in when you were in Freiburg, that I really remember that. So thank you very much, and thanks students. So I think there are a couple of exercises, I suppose, from the Apana or stock also, there were a couple of exercises. Please do those things. And please do also, there were about the reports, how you need to submit, and the WhatsApp, it's been displayed, the messages were given. Please do those things. And if you have any questions, please do put in the WhatsApp. So for today, that's all right now. Thank you all.
Thanks, everyone. Thanks, Kiran.
Thank you. Bye-bye all.